So this structure is called as the replication fork, a very favorite thing in board exam which had been repeated so many times to come in the board exams. So this is replication fork exactly drawn from the NCERT. Some additions I have done to mention the names of the enzymes here and there. Otherwise the basic skeleton of diagram is from the NCERT itself. This Y-shaped structure is called as replication fork. Remember, the whole DNA cannot be separated like this. It has to separate for a small stretch region. To stabilize a long piece of DNA once both strands separate would be a very much energy consuming process, and we need a large number of stabilizing proteins. We just learned about single strand stabilizing protein. So that's why only a small portion of DNA opens up, because once it is open, DNA replication will start and it becomes double strand, so it is stable. Now these are again stable. That's why small pieces will open, not the complete piece of DNA will open. Remember, there are two functions of deoxyribonucleotides given in NCERT. One is to act to provide the raw material, and second function to provide energy for bond formation according to NCERT. The next thing is formation of RNA primer. Now, what is the role of RNA primer? The problem is the enzyme required in this process is DNA polymerase enzyme, but there is a shortage in this enzyme. This enzyme cannot start the process of replication. It can only continue the process. So something has to start the process. So starting is done with the help of an enzyme called as primase enzyme, which will form a small stretch of primer. And please remember, primer is not DNA, but primer is RNA. Let's continue. So the next step is formation of the RNA primer. It's a short segment of RNA which is synthesized at the 5' end of the new strand before replications begin. 5' end of the new strand means opposite to the 3' end of the old strand or primitive strand. The enzyme primase will catalyze the polymerization of the RNA building block between A, U, G, C joined to form primer. Formation of the primer constitutes the initiation of the DNA synthesis, because DNA polymerase, same point, cannot initiate or cannot start the process on its own. Later the primer or RNA would be removed, and this gap will, because there will be U present there. U is found in RNA, not found in DNA. That's why primer has to be removed later on once the process has already started, and that gap which is created will be filled with DNA. Now the next step is base pairing. The two separated strands in the replication fork function as a template. The deoxyribonucleoside triphosphates will come and lie opposite to the approximate nitrogen base of the both exposed DNA templates according to the rule of base pairing. So if it is ATP, it will come opposite to the T, GTP opposite to C, TTP will come opposite to A, and CTP will come opposite to G, because I just told you activation. So they come in the form of ATP, but they will join in the form of AMP, GMP, TMP and CMP. So two extra phosphate present will go away with the help of the enzyme called as Pyro Phosphatase, another enzyme. So previous enzyme was phosphorylase. Now the enzyme name is Pyro Phosphatase, and the energy is released. This energy is utilized in the formation of hydrogen bonds between the free nucleotide, that means the new nucleotide coming on the new strand, and the nitrogen base present on the old strand or the template strand. Let me explain from here. Imagine a new strand. This is the new strand getting formed. This is the old strand present here. So if it is AMP present here, then it will be initially TTP, and then the two phosphates will be removed, so it will become TMP. So if it is AMP here, it will now become TMP here, and then the bond is formed, because we all know A and T will develop 2 hydrogen bonds. Similarly, G and C will develop 3 hydrogen bonds. So these bonds formation is helped with the help of the 2 phosphates which are getting lost, because they provide the energy for formation of these hydrogen bonds. So two role of deoxyribonucleotides. Two roles of this is what? First is to act like the raw material for DNA synthesis, and second is to provide energy so that the bonds can be formed. Now the next thing is elongation of the new strands. Now the adjacent deoxyribonucleoside monophosphate attached to the two strands get linked to form replicated DNA strand. It is catalyzed by the enzyme called as DNA polymerase III. There are three types of DNA polymerases in the prokaryotes. I'll be covering that soon. And eukaryotes have 5 types and require the presence of magnesium ion, ATP, GTP and TPP. So these is the cofactor. So these molecules are required for this process to go on efficiently. DNA polymerase III, along with the other polymerase enzyme first and second, has the capacity to elongate an existing DNA strand but cannot start its process. So they polymerize the nucleotide only in 5' to 3'. Now here one main thing to remember is that the new strand is always formed from 5' to 3'. In this case opposite, 5' to 3', 5' to 3'. So remember that when the enzyme will come, it will attach to the old strand's 3' end, and that means reading will always occur from 3 to 5 and addition will occur from 5 to 3. This is a base rule which you have to remember in biology or in genetics, that the reading by enzymes is always from 3' to 5' and addition is always from 5' to 3' direction, because they add them at the 3' end of the sugar molecule. As the two strands run in anti-parallel direction, because we have already covered in the helix model of DNA that the two strands are anti-parallel, so they provide different ends for replication. So because one strand is running from 3 to 5, the other is running from 5 to 3, so this strand will provide attachment here and this strand will provide attachment here. So two different positions for